Historically, microbiology has focused on one organism at a time, using culture-based methods to investigate each microbe's genome and morphology. Certain microbes, however, are unable to be cultured in the lab, and studying a microbe in a laboratory setting can often remove some of the contextual information contained in its native environment. Metagenomics is a branch of microbiology that directly interrogates the genomes of a microbial community in the context of its natural environment, without the need for culturing. Metagenomic studies typically use short-read sequencing to sequence complex genomes and metagenomes. However, with maximum read lengths of approximately 400 base pairs, assembling a contiguous genome and sequencing certain components of the genome, such as complex structural variants, can be highly challenging. Recent developments in sequencing technology have culminated in long-read sequencing, or third-generation sequencing. This technology produces much longer read lengths, with the longest reads to date spanning over 4 million base pairs, and has gained popularity in the field of metagenomics over the last few years. If you consider the assembly of a genome as a puzzle, short-read sequencing would produce a 1,000-piece puzzle of small pieces for you to assemble, while long-read would yield a 100-piece puzzle with much larger pieces. Not only does the short-read puzzle have more pieces, but to complicate things further, this technique cannot sequence the entire genome. This leaves gaps in the overall picture, introducing bias into the microbial metagenomes assembled from a sample. The larger reads produced by long-read sequencing allow for simpler assembly of complete genomes even from complex microbial samples. But, as you can see, even the larger pieces don't always fit together perfectly. These advancements in sequencing require new and improved methods for sample preparation and library prep to ensure that sequencing can be conducted on long, intact DNA samples. Sample preparation solutions offered by companies such as Zymo Research can be used in combination with long-read sequencing technologies, including PacBio's hi-fi sequencing technology to provide highly accurate long-read metagenomics. To find out more about the use of long-read sequencing for metagenomic studies of the microbiome, check out our in focus on the topic with Zymo Research over on www.biotechniques.com.